Hello, my name is Haley Britt, and I am presenting an article from the International Journal of Molecular Sciences. This article is called Analysis of miRNA Expression Profiles in Traumatic Brain Injury, TBI, and their correlation with survival and severity of injury, written by Consalvo et al. It was published September 2nd, 2024. Paper selection. One reason I selected this article was because of the recent publication date, published just about a month ago. Another reason I selected it was because I know how prevalent that TBIs are today, and they can be caused from anything from a hard hit during a football game to a fall from a ladder or a car accident. According to the Center for Disease Control and Prevention, there were over 69,000 TBI-related deaths in the United States in 2021. That's about 190 TBI-related deaths every single day. I wanted to learn more about the potential correlations between expression levels of these miRNAs and the severity and survivability of a traumatic brain injury. These authors hypothesized that miRNA biomarkers will be observed in cases with TBIs, and the expression of these miRNAs will depict differences between the severity and the survival outcome of the individual in these cases compared to the control group in this study. So looking at the image to the right, it is 3 MRIs that are shown. The first is an 86-year-old with probable Alzheimer's compared to a 14-year-old with a severe TBI, noting the similarities in the shadowing, compared to a 14-year-old with a healthy MRI, and noting the lack of shadowing. So some background information. What is a traumatic brain injury? So according to the CDC, it is an injury that affects how the brain works. It can be caused by a bump, blow, or jolt, or a penetrating type of injury such as a gunshot wound. So in the journal article, the study that was conducted was on 40 case subjects that were deceased with a TBI, and age ranging from 12 years old to 65 years old. And these are the test cases that were used. And then there was 10 control cases that were studied. They were deceased suddenly from a cardiovascular, non-traumatic causes, and these range in age from 24 to 65 as the control group. The miRNA data that is collected at the different expression levels will be compared and analyzed to determine how the TBI affects the MRI in the brain compared between the two groups. So a summary of methods. For RNA extraction, there are 4 steps. The first is deparaffinization. Xylene and ethanol were added to the tissue sections and removed the wax and exposed the RNA sample. And for digestion, a PKD buffer and proteinase K were added to digest the cells and break down. And then during DNase booster buffer and DNase stock were added to remove any of the contaminants that were going to affect the sample. And 4, they were purified in the centrifuge. The sample was purified using a centrifuge machine to separate out the RNA for extraction. And then for the RNA assessment, a concentration of the RNA was assessed using a nanodrop spectrophotometer, and it was processed by a cDNA synthesis kit for reverse transcription of the miRNAs. So results. So figure 1 is going to be in the upper right corner, and it was first used to divide the case subjects into 4 subgroups based on the time between the initial injury sustained and the time when they passed away. And so the first group is going to be the fatal TBIs, which is no survival, and those are going to be in gray or not show up at all. And then the next group with the short survival time of less than a day is going to be in the darkest blue color, and it's going to go up to the first line of less than a day. And then in the medium blue color is going to be a medium survival rate, the middle survival rate of between 1 day and 7 days. And then in the lightest blue color is going to be the long-term survival rate of more than 7 days. And for figure 2, this divided the cases into subgroups based on the severity of the trauma using the AIS score, or the abbreviated injury scale, and this can be used to describe the severity of the trauma that the patient has suffered and place them into different subgroups based on this. So in these cases, an AIS score of 1 or 2 is considered a mild or moderate TBI, and a score of 5 or 6 is considered critical to fatal. So an AIS of 1 might present with symptoms like a mild brain injury with no loss of consciousness or whiplash and something like that, but an AIS of 6 might present as damage to the brainstem and brain death, which is much more severe. And this is represented in the figure 2 on the bottom right, with the light peach color being on the bottom, going up to the 2 line, which is the AIS of 2, and then the red going up to from the 2 to 4, the AIS of 2 to 4, and then the dark maroon color going from the 4 to the 6 in some of these cases, representing it goes through 1 through 40, representing which patients sustain these injuries for both of these figures. So some more results. So this is figure 3. And in figure 3, the box plot data represents the miRNA expression levels in the case compared to the control group. So the orange box plot represents the 40 case study participants, and the blue box plot represents the 10 control group participants. So what does this data tell us? This analysis revealed an upregulation or a higher expression of miR16-5P, miR21-5P, miR130A, 130P, and miR155-5P. So that is an upregulation or a higher expression that you can actually see if you look at box plot A, C, E, and F, as the orange is going to be higher than the blue that's expressed in the box plot. And then a lower expression was detected in miR23A and 3P, and that's going to be shown in box plot D. When you look at that, the blue will actually be higher than the orange in this case, which shows a downregulation or a lower expression. And then in box plot B, there was no significant variation in the MRI-19A3P between the control case and the case study. So this is figure 4. Figure 4 is showing a clustered heat map with the 5 miRNAs that showed the variation between the control group and the cases. So the left figure in blue shows the clustered heat map on survival time, and the right figure in red shows the clustered heat map based on the severity of the injury that is based on the AIS scale. So a cluster map is just a way to visually show the correlation between the miRNA and the different groups. So the darker the color, the higher the expression level that there was for that miRNA at that time in that group. So if you look on the left-hand side at the survival clustered heat map, the short survival TBI in the second column from the left, at the top row, it has the darkest color, which shows that that has the highest expression for the MRI-16-5P for that miRNA. But when you look at the severity clustered heat map for on the right side, the darker color in the critical to fatal TBI in the far right column, the two darker colors at the top 2 rows, those are shown at the MRI-21-5P and the MRI-16-5P. So these clustered maps allow us to make quick analysis and understand where the differences are showing up, like the short survival time TBI and at the MRI-16-5P, which is approximately a 6-fold upregulation in the short-term survival subgroup, higher than expected. And so that's definitely a difference that was not expected. So you want to analyze why that happened and understand it. So main points and conclusions. The study identified 5 different miRNA that may directly correlate to the damage of brain cells, populations, and neurodegenerative mechanisms in the brain. There are high potentials for therapeutic uses of modulated RNAs. In conclusion, these results suggest that miRNA analysis could become an essential tool in forensic investigations in the future, providing more accurate and detailed information about the nature and extent of TBI. The predictive power in relation of the timing and severity of trauma could ultimately contribute to the definition of the circumstances of death. So, the significance of these findings? Strengths. So the first strength that I noticed was the great visual representation of the data that was gathered during the study. The cluster heat map and the box plot graph were able to present the most important findings without having complicated keys or figures underneath. They were able to show that this was important, and if someone was looking at it, they would notice that, like, especially in the box plot graph, that they would know that this has a higher expression simply because the box was charted higher than the other one. And the next strength that I noticed was that they had very detailed information on the members of the case study. This included age, sex, cause of death, traumatic mechanism of death, type of brain injury that was present, post-mortem interval, and whether any surgical treatment was performed on the person before they died. Weaknesses. So I do include case study size as a weakness, which included 40 of the case study participants and 10 of the control cases. So this is 40 of the cases of people that died with a TBI and 10 control cases of ones who died with a cardiovascular incident. 50 is a lot for a human case study, but I would like to compare the data against a larger sample size and see how it compared to see the expressions of the mRNAs and see if they matched up with against a larger sample size. The second weakness I chose was the method explanation. So the method explanation did take a lot of dissecting to understand the techniques that they were using, and a simplified explanation would make it a lot easier for more people to be able to comprehend and make it more accessible to other people that weren't necessarily as involved in the science realm but wanted to understand the process or were interested in this type of research. And then the third weakness that I chose was the color selection on the graphs, especially the two bar graphs, which were figure 1 and 2 in the beginning of the presentation, because they used a lot of the same colors and just were different shades of the same colors, and that could be hard for people to tell the difference between, or hard to determine which one correlated to the different item in the key, and it would make it a lot easier if they would just choose varying colors to depict the different things they were trying to show. But overall, the information that they provided has potential to be very significant. So if the identified mRNAs can be modified to be therapeutic in some way, it could increase survivability of these types of injuries or improve the quality of life for many people out there. And these are my references. Thank you so much.